Hello everyone, in this particular video we are going to discuss about the sulfonamides, which are antibiotics. As you know that antibiotics are the agents which act against the bacteria. So sulfonamides are the agents which act against the bacteria, right? They act against the bacteria, show antibacterial effect. So sulfonamides were the first effective AMAs, that is they are the first effective antimicrobial agents used in humans for bacterial infections. They were the first effective antibiotics used in humans for bacterial infections, right? Also, they are the derivatives of sulfanylamide, that is para-aminobenzene sulfonamide. And the point to remember is that they are the first synthetic compounds, that they are the first synthetic antibiotics, right? So coming to the structure of sulfonamides, it contains a para-aminobenzene ring. As you see here, this is a para-aminobenzene ring, right? They contain a para-aminobenzene ring, and they have N1 substitution, that is SO2 NH2, for the activity, for the solubility, potency, and pK property, right? This N1 substitution is required for the solubility, potency, and pK property. Also, the N4 free amino group, this is the N4 free amino group which is responsible for the antibacterial action, which is responsible for the bacteriostatic activity, right? This is important for the antibacterial action. So coming to the classification of sulfonamides, sulfonamides are classified into systemic acting agents, local acting agents, and sulfasalazine, which acts both locally and systemically. Okay. So under systemic acting agents, we have short acting drugs, long acting drugs. Intermediate acting drugs and long acting drugs. For topical acting agents, for local acting agents and topical action acting agents, we have sulfa-acetamide, silver sulfadiazine, and mefanide. This sulfa-acetamide sodium was actually used for the ophthalmic purpose. And see, the short acting drugs, they act for 4 to 8 hours. They show action for 4 to 8 hours. The example is sulfadiazine. The intermediate acting drugs, they act for 8 to 12 hours, and the example is sulfamethoxazole. For long acting drugs which show action for 7 days or more than 7 days, they examples are sulfadoxine, sulfamethoperazine. For the special purpose sulfonamides or sulfa-acetamide sodium, which is used for the ophthalmic purpose, and sulfadiazine, silver sulfadiazine, sulfasalazine, and mefanide. Coming to the mechanism of action, see, the PABA, which is a para-aminobenzoic acid, it is a precursor of folic acid. It is a precursor molecule. A precursor molecule means it is an important molecule for the formation of folic acid. In bacteria, for the formation of, in bacteria, for the formation of folic acid, we require PABA, that is para-aminobenzoic acid. Okay. Next, as there is a formation of folic acid from PABA in bacteria, there is growth and multiplication of that bacteria. Okay. So these sulfonamides, what do these sulfonamides do is that they are the structural similar to PABA. They are structurally similar to this PABA. As PABA is required for the formation of folic acid, these are structurally similar to the PABA. They competitively inhibit the folate synthase enzyme. They competitively inhibit the folate synthase enzyme and prevent the formation of folic acid. As there is no formation of folic acid, it produces bacteriostatic effect, that is it inhibits the bacterial growth, right? It inhibits the bacterial growth. And also it is important to know that these sulfonamides, they are not effective in presence of pus, as because pus is rich in paba, purines and thymidine. Okay. In mammalian cells, see, in human cells or in mammalian cells, we generally do not synthesize any folic acid, right? We take folic acid from the diet. So when we take sulfonamides, it doesn't cause any effect for the host cells or for the mammalian cells or for the human cells. It will not cause any harm. But however, the inactive metabolites of the sulfonamides, they have some side effects. See, this is a para-aminobenzoic acid, that is the PABA. It is important for the formation of folic acid. Okay, all this will be happening in bacteria. In bacteria, it is important to know that in bacterial cell this process is going to happen. Okay, in the bacteria which has affecting that particular human. Okay, in our body, if there is a bacteria, in that bacteria this is a normal metabolic pathway in bacteria, right? So when we take the PABA, it will be converted into dihydrofolic acid in the presence of folate synthetase, right? In presence of folate synthetase, PABA is converted into dihydrofolic acid, and after that this dihydrofolic acid in the presence of folate reductase it is converted into tetrahydrofolic acid. Okay, okay, why folic acid is important to bacteria is that this tetrahydrofolic acid is important for the formation of DNA, RNA and proteins, right? This tetrahydrofolic acid is important for the formation of DNA, RNA and proteins. DNA, RNA, proteins are the compounds which sustain the growth of bacteria, right? When we inhibit the formation of this tetrahydrofolic acid, then there is no further development or further growth of the bacteria, right? For that reason we inhibit the folate synthetase and the whole process will be inhibited. Okay, for that, the sulfonamides, as the sulfonamides are structurally similar, they are structurally similar to PABA. They are structurally similar to PABA. They inhibit the PABA. They inhibit the folate synthetase. By resembling PABA, the sulfonamides, they inhibit the folate synthetase, and there is no formation of dihydrofolic acid and tetrahydrofolic acid, and there is no formation of DNA, RNA and proteins of the bacteria. Hence, the bacterial growth will be decreased. Okay, so that's all for this video. Thank you.